Welcome, everybody. Good afternoon. Welcome to webinar number 8 of Platomics IVD Ready program, which we have given the title IVD Ready to Go. My name is Simone Latkulik. I'm leading the customer success team for labs at Platomics, and I will guide you through this webinar today. We have special guests invited. Let's have a look at the agenda and our speakers. Professor Dr. Volker Spitzenberger, Regulatory Affairs expert at the University of Applied Science in Lübeck and group leader of Clinical and Regulatory Affairs at Fraunhofer Institute, will talk. He actually advises international organizations such as the World Health Organization and the European Union in the field of quality management, quality assurance, and regulatory affairs. Professor Spitzenberger will give us a preview on the future ISO standard 5649 for medical laboratories. That's described concepts and specifications for the design, the development, implementation, and the use of laboratory developed tests. Also, he will talk about current transition timelines of the IVD medical devices. Professor Dr. Ronald Simon from the Institute of Pathology, head of the section of molecular pathology at the University Medical Center Hamburg-Eppendorf, will talk about the implementation of the in vitro diagnostic regulation in a routine diagnostic molecular pathology lab. Last but not least, Andreas Oberleitner, Chief Regulatory Officer at Platomics, will give an outline of the PlatoX platform. You can ask questions in the QA section during the webinar, and we will try to answer them by the end if we have enough time. Otherwise, we will come back to you via email if you ask questions non-anonymously. Professor Volker Spitzenberger will now give his contribution. Professor Spitzenberger, please.
Welcome to everyone to this midday or afternoon session on LDTs, in-house IVDs, and I thank you so much for the invitation. As already announced, the preview on the future ISO 5649 on laboratory developed tests will be the first aspect of my short presentation, and then the current transition timelines for IVD medical devices, of course, with a strong focus on the in-house IVD medical devices. So what was the background and rationale for this new or upcoming ISO on laboratory developed tests? Well, the initiative actually took place from the European perspective, and was actually an initiative that was strongly, let's say, triggered by the European IVDR. But nevertheless, the background was, of course, a background that was set by regulatory requirements from an international point of view, from an international context. And I would just quickly highlight some of them. For example, most current, of course, the new rule on laboratory developed tests as issued by the US FDA, where the main focus is that IVD should be manufactured, or IVDs manufactured by a laboratory should generally fall under the same enforcement approach as other IVDs that are commercially produced and placed on the market, which is the current perspective of the United States Food and Drug Administration. On the other hand, we have the European initiative, as already shortly announced, where we have the major focus on, let's say, the fulfillment of the essential requirements for safety and performance of the IVDs that are in-house manufactured or developed and modified, and then used within one health institution, where certain rules of the regulation should not apply. Of course, you have already heard a lot of this aspect and of all these requirements, and also of those requirements that do not apply for in-house IVD medical devices in these webinars. Another perspective is the one from actually Australia. Australia actually has a very let's say, mature and quite strict regulatory framework for IVD medical devices. And last but not least, Health Canada, on the other hand, actually communicates that Health Canada is the official, let's say, competent authority that is competent and responsible for the regulation of health products in Canada. They say, okay, laboratory developed tests are not subject to statutory regulations on medical devices in the, let's say, on the national perspective, on the national field. So all this has to be considered when talking about requirements. On the one hand, regulatory requirements, but on the other hand, also, let's say, normative requirements, state-of-the-art requirements from the medical scientific point of view for this kind of examination procedures. That was the regulatory point of view, the medical scientific point of view with regard to what should be done according to the state of the art for the RDTs, and to, of course, ensure their performance, their safety, their quality has been communicated by many publications during the last years. Here is only an overview on some of them that have related to, of course, the European IVDR, have related to ISO 15189, have related to other standards, and maybe also other regulatory aspects. So this is all about the background and rationale for the project that has been initiated by the International Standardization Organization some few years ago. And that actually has started in 2021, in December, when, as I said, triggered by the European IVDR, but not only, of course, the IVDR, also the overall background that I try to summarize here. The committee, the ISO Technical Committee 212, which is dealing, of course, with IVD medical devices and with also quality and competence in the medical laboratory, they decided to actually register a project on concepts and specifications for the design, development, implementation, and use of laboratory developed tests. And so that project then has been developed step by step, of course, and we are now in July 2024. A DIS vote, a so-called DIS, that is a draft international standard, had been prepared step by step. There was a vote on it that has been finished in March 2024, successfully finished. That means we can go as a committee of step forward, which is then the so-called final draft international standard, with the overall objective to finalize the standard within this year. Of note and of interest, of course, that standard will not only be an international standard reflecting the international state of the art with regard to the requirements or specifications for laboratory developed tests, but it will also be a European standard. So by a certain procedure, the standard was developed in parallel as an ISO standard and as a European standard. The drafting committee is actually, well, all the co-authors that have been contributed to this future standard. And I have, of course, implemented or integrated this slide here, of course, to thank all the contributors on the one hand, but also to make clear that this standard had or has a quite a huge contribution out of the European stakeholders. So although it is an international standard, and we have really very much appreciated contribution from United States, Canada, also Asian countries, Australia, and so on. Also, as you can see here, Japan, for example, as an Asian country. But we have really a huge contribution from the European stakeholders because the, of course, the interest in this topic is especially high under the background of the European IVDR. So what is the scope of that standard that is related to the specifications? It outlines general principles and assessment criteria by which an RDT shall be designed, developed, characterized, manufactured, validated, and monitored for internal use by medical laboratories. It also includes all those types of laboratory developed tests, and I will come to that point later, that are based on commercially available IVD medical devices, but that are modified to a certain extent by a medical laboratory. It, however, does not focus on the how, it focuses on the what to do, not on all the tools and all the details how to achieve to fulfill the requirements. That is something which is important because it is an ISO standard. It is not a technical specification. It's not a technical report that usually are intended to describe standard or to describe how to fulfill a standard. That's not the idea of this. Here we really talk about the specifications and the requirements. We do not cover, of course, commercially available and commercially manufactured products, and we do not cover in this standard research use only devices, although of course the principles that are set in this standard might be useful and might be applicable also to this kind of devices. Here you can see an overview on the future ISO 5649, which is currently, as I said already, in the procedure to develop it from the DIS to NAFDIS. You can see here major topics on terms and definitions, which are very important. I will come to this point later on again. You have a section on general requirements, and may I draw your attention to that chapter 4.1, rationale for laboratory developed tests, as you can see here. We had a big discussion in the project group on what to implement here and what to include here. You might already see the context and the connection to the so-called justification. That is one of the points out of the European IVDR, justification for the use of a laboratory developed test. And the project group decided from an international point of view to say, well, we don't have to justify any kind of use of a examination procedure in the laboratory, but however, a rationale would be very important. So what are factors that might even urge a laboratory to develop an in-house IVD or to modify a commercially available device in order to make it a reality. So next section will be on the requirements for the design and development of laboratory developed tests. So all about intended use, scientific validity, risk management as a very important part, and of course the association to the essential principles for safety and performance, which is an international term for what we call in the European Union the essential requirements for safety and performance. Then the performance evaluation phase takes quite a high focus on in the standard, and of course when performance evaluation, which is a procedure that relates to validation, verification, and risk management, then took place, we come to the next section and to the next stage of the life cycle of an LDT, which is the implementation, monitoring, and finally the retirement of the test. So all this, and you can already see, is actually covered in a so-called life cycle. So the possible, maybe not all of the life cycle phases might be applicable for any lab, but the possible life cycle phases of an RDT are actually pointed out that standard, from the rationale to the feasibility assessment up to so many steps and the final retirement. And as you can see here, these stages or phases are then always, let's say, of course, connected or associated to the different sections of the standard. So relevant terms, which is very important because so far and up to today, there is no internationally agreed, let's say, and internationally agreed term and consensus on that term. So that was a high debate, as you can imagine, about what really is a laboratory developed test, how should it be called, is a laboratory developed test the same as an in-house IVD, and so on and so on. So laboratory developed test is used as a synonym for in-house IVD, what is the preferred term, maybe in Europe and maybe in Australia, but not in many other countries in the world. You can also call it a laboratory-developed examination or a self-developed method. And it is a test or examination which is designed, developed, manufactured, or modified and used within a single laboratory or laboratory network to carry out testing on samples, where the results are intended to assist in clinical diagnosis or be used in making decisions concerning clinical management. Also, the definition points out and describes quite in detail the different scenarios under which LDTs or in-house IVDs might be produced or used within the medical laboratory. And of course, of note and of highest interest might even be the scenario 4, where you say the LDTs are LDTs when they are used for a purpose, or when the IVDs are used for a purpose other than the intended purpose assigned by a commercial manufacturer, including significant changes of the IVD. And that brings us again here to this definition that points out what that kind of significant change with regard to intended use, for example, could mean. I cannot go by time reasons and all the details of these definitions. I just would like to give you some short overview, of course, what that standard can bring out for all of us in the laboratories, of course, and what it tries to bring up as clarification. Significant change is, of course, a very relevant term. That's why I would like to bring it here again as one of the examples of those terms that are defined in the standard. It's a change that could reasonably be expected to affect the quality, safety, or performance characteristics of an LDT for its intended use. So a significant change includes a change to any of the following, and of course, intended use of the LDT is one of those changes that are to be categorized as a significant change. Changes in the manufacturing process, facility, or equipment, in the quality control procedures, or of course in the design of the test. The term, or let's say the fundamental basis for this kind of the definition is in essentials two documents. The one is brought out from the Canadian guidance document for the interpretation of significant changes, brought out or published by Health Canada. The other one is the European document, the MDCG 2022 document on the significant changes that are related to so-called legacy IVD medical devices. Another relevant term might be the performance evaluation, because we talk a lot in the IVDR about it, but might be relevant to know what the performance evaluation of an LDT actually is. It is understood as a comprehensive investigation process for the examination procedure. Data are generated from validation verification studies, and performance evaluation includes activities of risk management and supports the demonstration of the conformity of the LDT with the relevant or applicable essential principles of safety and performance. So that might bring out some clarification on the international and European level with regard to these terms. What does the standard actually require? Well, it has a strong focus on, as I already said, the QM system and the risk management system. It actually relates to appropriate QM system. It does not say, well, you need to be in conformity with ISO 15189 or ISO 13485. This is up to the legislators, to the regulators, but it actually points out which QM systems are appropriate, and that the QM system should cover all stages or phases of the LDT life cycle, which is probably the most important thing. It has aspects on the transfer of devices, which is of course a very hot topic in the European Union, as we all know. According to the IVDR requirements, Article 5, it talks about the control of subcontractors. The risk management system is proposed or suggested as, of course, a cyclical risk management process throughout all the phases of the LDT life cycle, and it The standard also introduces a so-called risk differentiation. So that means we have to talk about the risks related to the intended purpose and indications for use, and on the significance of the results produced by the LDTs. That is, of course, a topic that relates to the risk classification that is obligatory for in-house IVD medical devices according to the European IVDR. But this is, of course, not the only jurisdiction that requires risk classification. There are many others, but it's also, of course, a link to the European system. The whole implementation and monitoring of LDTs is also following a kind of cycle. And maybe it might be of note that the title talks about implementation because we had, or the project group highly debated the wordings about production or manufacture. So people did not want to say that a lab produces something really. So they said, okay, we are going to design and development, maybe to modify and to implement. So which is actually meant then that after this design and development phase and after you have done your performance evaluation and your and have implemented your risk management process, you're trying to of course to implement that LDT in your laboratory. Timeline is already said. We are currently now here at the final stage of the DIS towards the FDIS. So the FDIS is already under preparation, which will again need another vote, so a ballot. For that FDIS, which will only refer to editorial comments of this document so far. So it's really now a mature document, and the idea is really to publish it during this year, simultaneously as a European and ISO standard. Some few more remarks on the transition timelines. So this was the first part, and some few aspects only left to the transition timelines for IVD. Here in this slide, you can see the major, let's say, regulations, of course, for the medical devices in the European Union, MDR, IVDR, brought on a timeline. And we all know that they were amended several times, that they were changed, amended. And the last amendment is quite interesting. That is actually dating from January 2024 as a proposal, and that proposal, of course, has been proceeded for several months. You can see here the official European communication from the European Commission dating back from 2017 until today with that legislative proposal that I already mentioned, and that gives further extension of the transition periods for commercially available IVD medical devices in dependence from their risk class on the one hand, but that also includes, of course, another idea on a further transition or extension of the transition period for one special aspect on LDTs. And that's what I would just quickly like to refer to again. Here you can see again on the left, of course, the summary of the requirements according to Article 5.5 of the IVDR. And on the very right, you can see, let's say, the major aspect that a lab has to consider and has to fulfill, as you all know. And above those major elements, you can see the deadlines. The deadlines, of course, that the deadline that most currently has already passed is the one on the 26th of May. That is about a little bit more than 4 weeks now ago, with regard to the QMS, to the Class D documentation, to the EU, or not the EU, but maybe let's say the Declaration of Conformity and the product monitoring. Which has actually now achieved that deadline. Of note and of interest, the justification already mentioned at the very beginning of my short presentation, that justification for use is now further extended from May 2028 to end of December 2030, which is something which might be not so much in the focus of the stakeholders of that amending regulation. The regulation itself, actually, to my understanding, is already signed by the European Council, but it has not been published. That proposal from January, that is the amending regulation, has not yet been published in the European Journal. Why, I don't know. I think we can all probably expected this will be done in the next few days or in maximum weeks. Which will also mean that the well-known MDCG guidance document 2023-1 on Article 5.5 of MDR and IVDR will have to be changed because of the new deadline here that refers to the justification for use. So by this, I already thank you so much and will finish here my small presentation, short presentation. Thank you for your attention, and I'm happy to expect any comments for the discussion. Maybe later on. I don't know how far we have the time, but we should move on probably.
Thank you very much, Professor Spitzenberger, for this very interesting contribution and the preview on this new ISO standard, and also giving an elaboration on the IVDR current timelines. I would like to announce Professor Dr. Ronald Simon from the University Clinic Medical Center Hamburg-Eppendorf. He will give a presentation on the implementation of the IVDR in his diagnostic laboratory. So please, Ronald Simon, I would like to hand over to you.
Thank you very much, Simone. It's an honor for me just to talk about our first experiences here with the IVDR, and a few words to me. So as you can see, here's a microscope. I'm working in a pathology institute in a routine diagnostic molecular pathology lab. So I'm no expert for regulations, IVDR, or anything in this place. I'm just a user of molecular diagnostic tests. And you see it's a large hospital here in Hamburg. Our Institute for Pathology, we're quite large. So 128 technicians, more than 50 pathologists, molecular pathology division, research division, bioinformatics, all you like, and more than 100,000 patients that we serve per year. So just to say, my job is to provide molecular diagnosis to our patients. And this is just an overview about the test assays that we have. For example, in the pathology lab, you have antibodies. So 285 antibodies. We have fluorescence in situ hybridization assays, 83, PCR-based 38, next-generation sequencing, 5 assays, and some other small things. Now, and we have the IVDR. So, and this is 157 pages and 14 annexes. Me as a lab person, I'm looking a little bit like that, because the question that came immediately up years ago, so when the IVDR came up, was, do I really have to read it all? And who helps me to understand it? The best question probably, does somebody have a template? Because you see, we are used to, if you know someone who has already done the work, just go there, see what they have done, have a template, and do it exactly the same. Probably that's a very successful way. And finally, but what should we start with here? So just to tell a little bit from our experience, step one was see what you have. And this is simply said, if we look at our test, then we found, yes, in fact, we have fully CE IVD marked tests. A lot of antibodies, that's nice, so they are CE marked. The automated stainers that are used with these antibodies, they are also IVD CE marked. That's perfect. We have even an NGS assay with a CE mark and a computer attached to it with a program which is fully IVD CE, so this is on the safe side. Then there is another bulk of tests, I call it a little bit CE IVD, and this is very nice. So when we listen to Professor Spitzenberger, he very nicely explained that this is everything where, yeah, for example, the FISH probe itself that has an IVD, a CE IVD label, but we use then our own buffers, our own hybridization systems, and so that's then obviously something we need to take action at. And all the rest that is not CE IVD marked at all, so what can we do? Step 2 is then, in my eyes, just categorize. So category number 1, fully CE-IVD, we are fine with that. Category 2 is everything which is a little bit. And category 3 and 4, so there are 2 options. Either there is an IVD alternative available, then you can think of buying it, or it is not available. But here it comes to, we have to make an in-house test. That's step 3. That's the action that we have to take here. And the question is, how can we do that? So prerequisites. Professor Spitzenberger showed several slides on that, and that was extremely interesting for me to learn that there's so much you have to look at. I picked only a few items here where I think they are maybe most important for us as users. So number 1 is for your own lab only. So of course we must not distribute that, we must not sell it, because otherwise we become CE-IVD manufacturers by our own, so like a company. That is not allowed. We have to have a QM system. This is fine because we have that, so we do not need to take any care about that. Justification, we learned, it's not from May, sorry, it is December 2030. Performance data available. Here I become a little bit unsure of what it is, and we saw in Volker Spitzenberger presentation that might be not so easy. Risk classification, I tell you, when I read that, I had no idea what that means. Then that's the declaration that the test fulfills basic safety and performance requirements, and then anything else. Maybe I overlook at least 10 points that are important here. So that's okay. The justification that comes back to the question, what is good for? Just an example. When I thought about that, we have an example of an NGS assay, 500 genes. The intended use, I would say, like the 500 genes NGS assay is used to search for mutations and gene fusions in about 500 genes on the DNA and RNA level. That would be, from my basic feeling, the intended use of that. That makes the justification easy because there is no such test available with CE-IVD marking. So even there we have a justification. But when it comes then, sorry for the performance, risk classification, declaration, performance. This is what we have. And maybe every molecular pathology lab or molecular lab has. At some time, we validated our assays. Then you have a standard operating procedure of a test. You have a validation report of that assay, of the devices, of reagents that we use for that. And this is all fine. We have that documentation, but still the question is open, is that enough? Do we need more? And if we need more, what do we need? Risk classification. Declaration, I did not know what I should do here. I was hoping that someone could tell me just what to do here. And anything else, again, what things do I probably overlook? Coming back to my, so how I looked when I thought about that. Does somebody have a template? And now here comes the good news. Yes. I was really happy to see that Platomics website where there is an IVD ready for your lab system. And the good thing about this, it was free. It is free for the labs. You just need to request access. And I would just like to show you what we did so far and how handy it is, how we used that. That is when you go to that server, to that website, you just have to log in there, and then you come to a products page. You can already see I entered some of the products, so the workflows, the tests that we did here. So the first thing that I learned, and this you can see here with the workflow, very interesting. So I always thought before, If we have something like the 500 genes assay, it's only that 500 genes panel that we probably need to make to an LDT. But no, it's more. There's a workflow behind it. So it means there are devices and reagents for DNA extraction, for RNA extraction, PCR machines to generate libraries, and all these different kind of lab equipment and reagents that all make up the entire workflow. And that is quite good, because if we tip on the here on that workflow and see what is behind that, there is a list of things you can now look after and fill it in. You see there are all green bullets here, fortunately, because we have just done that for that essay. I have to apologize, it is in German, but however, you should ever use it in the language of your lab, because then it's easier for everybody to understand, and most of all for the people who are working in the lab. So you just go through it from point to point, general information, or the intended purpose here, the risk classification, workflow, the components that you use in the workflow, like devices, buffers, reagents, the basic safety and performance requirements. Here I can look it up, what it is, and get help. I did not know about that. It's the environment where my tests is. I would never have thought that this is important, but obviously it is. And here you get a catalog of questions you can answer to make sure that you have properly documented which environment it is. Environment means something like temperature, humidity, all these things. And the performance characteristics. Let's look at it point by point. For example, here, the intended use. There opens a dialog where you can tick, what is it? So my 500 genes test we are talking about, obviously it uses an instrument, that's a sequencer, a kit, this is the 500 genes panel that we buy from a commercial vendor. Yes, there are also containers where the samples are in, reagents, software, all this you can hit. What kinds of analytes do you here have, and other things. So a few questions, so a few pages of questions, but then it gives you as the result the intended use. And when I thought of my first sentence, I had like, the panel is there to search for mutations in 500 genes. That was quite simple. But here I have the feeling that's quite a more sophisticated and probably more correct intended use of my product. And I did not generate that by myself. I just answered questions, and then it comes out what the intended use is. So it puts it automatically together. So panel 1, NGS workflow, is a semi-automated workflow consisting of an instrument, software, kit, reagent, and sample containers, and is intended for in vitro analysis of DNA and RNA for qualitative detection of mutations and fusions of about 500 cancer-related genes. The panel 500 genes workflow provides information to identify clinically therapeutically relevant DNA RNA alterations in individuals with tumor disease suffering from advanced tumors with limited conventional therapeutic options, and is used by trained laboratory technicians, pathologists, healthcare professionals, and physicians within a healthcare facility. That sounds good, and this was generated by the system. Risk classification. You answer questions in these dialog boxes, and here's the result. Risk class based on the applicable rules. The following risk class apply. It's class C. Classification rules. Rule 3 is applicable in particular. The following subcategory, 3K, management of patients suffering from a life-threatening illness or whose condition is life-threatening. the system generates this answer. I looked it up because I was just curious in the IVDR. And yes, I found that molecular pathology assays and diagnostic immunohistochemistry assays, these are typically all class C. So there will be probably no other risk class in our assays that we use, because these three rules you can find: use as a therapy accompanying diagnostics, use for cancer screening, diagnosis or staging, and 3K, management of patients suffering from a life-threatening illness or whose condition is life-threatening. So that fits nicely with what you would expect if you have gone very carefully through the IVDR and found these rules in the appendix. The general safety and performance requirements. This is really a piece of work, but again, the system makes it easy. There's a wizard for that, and the wizard means you're getting a catalog of questions. And I tell you, these are more than 200 questions that you need to answer. So this is worth of taking 1 or 2 days time, a good cup of coffee, and then you go through all of these questions here. So they are all explained. So if you tick here somewhere, you will see what the requirements are. You have an overview about the instructions here, the documents, and you have even a description why this is important and how you can get help to find the answer to your question. Very interesting, the documents that you need for that, and I would have never known what kind of documents I would all need. So it is asked for, and you can choose from a database. So these are your own documents that you have already made in the first step. They all go into a database, and then you can search that database and just tick and select the documents from that database. Once you did it for a few questions, you get used to it, and then it's really a self-learning system. So you have access to all your documents and just hit when it fits to your answer. Yeah, this is just an example of the documents that we have, for example, in this database. So it grows and grows with every step you go through the system. What comes out finally, so you generate then your general safety and performance requirements. That's in fact an 88 pages document where all these information are given about what's the application, what are the product information, what's intended use, the description of your test, the answers to all these questions, and the system just selects these questions that apply for your test based on the answers that you give to the questions. So very handy, 88 pages. Performance evaluation. As said already, we have standard operating procedure of our test. We have a validation report of the test, of the devices, of the assays. Anything else, you can find it all here. So you go through these questions for that, and then you get as an answer: the performance evaluation confirmed the state of the art of the project, the performance of the product according to its intended purpose and requirements. The product meets the standard general safety and performance requirements, and the overall risk-benefit ratio is acceptable. So also again generated by the system. Anything else? Well, this is the only document, in fact, that we generated by ourselves, and we just made a page where we have the declaration that we can put on our website. Nor the relevant documents, we just have it in a folder, and if it comes to the point that an authority comes and wants to see it, we have it. Very nice features, again, is you can, that is the copy button here. So once you have made one workflow like that 500 genes workflow, I just copy it, generate a new workflow because we have also a smaller 60 genes workflow. And it's completely easy because you just adapt to the 60 genes here and all the questions for the general safety performance requirements. This all stays basically the same. You just check if it's correct, and within a few minutes you can generate the third and the fourth test. And so the more tests you have, the more templates you have generated by yourself. You just copy or duplicate them and adjust them. The requests component button, that is also a very nice thing because for specific components that you use, for example, for that kit that we purchase from a commercial vendor, there are, from the vendor of that kit, there are some information like safety data sheets or just guides how to use that. These documents are there, not for all companies, but I think Pratomics did a good job so far. And many companies that produce devices, reagents, or other thing like software that is typically used in this text, they have uploaded their documentation to the database. And this is very helpful because then you have it directly from the vendor. You can add it to your assay and do not need to take care about your own validation steps for devices or reagents. If a component is not there, you have a request button that goes to Platomics, and they try to get in contact and touch with the vendor of the product and see and ask the vendor if they would also put their products in the database. Well, here at the end, I think it turns out, yes, you have to read it. There's no way around it. But we have help how to understand it. So there are a lot of answers in the system, a lot that you can look up. Yes, there is a template. I found it really beautiful. Once you get used to it, you can work with that, and you can start with it really right away without having too much knowledge on the IVDR. Summary. For me, it was exactly the kind of template I was looking for. It's a step-by-step how to do it. Software handling is quite easy, and just a questionnaire style is really good to use. Some answers require knowledge of the IVDR and specifically its language. Sometimes it's hard to understand, but at least you know what you're looking for, and you can then find answers. And since I now have the email address and telephone number of Professor Spitzenberger, that will make life then much easier. Similar workflows or tests can be quickly generated by duplication, very handy. And important point, no more worries about did I forget something, because it is in the program, it just asks you. Nice, free for academic labs. Well, some things below. Component database is great, but still under construction, though that, of course, would be very nice if more companies add their devices there. And so far, to my knowledge, there has been no review of the generated documents by the authorities. So let's hope that when somebody comes at some time to our lab and wants to review these documents, that it's all okay. But having said that, it is really a great work, and I feel very comfortable with that and can only ask you or suggest just try it. It's quite easy. Well, thank you so much. That was it, the experience from our lab, and I'm happy to take questions.
Thank you very much, Professor Simon, for this comprehensive presentation, how you implemented the IVDR in your lab. Thank you very much. I would like to hand over to Andreas Oberleitner, Chief Regulatory Officer at Platomics. He will outline the Plato X for us. Andreas.
Thank you very much, Simone. Thank you also to my previous speakers. It was great to see the experience, how the tool is already used in health institutions. Yeah, due to time constraints, I will only very quickly go through a few slides just to sum up what we have seen so far. I would like to take the opportunity also to have a look back in what already happened in the IVDR domain so far. Yeah, it had been mentioned already by Professor Spitzenberger, and as we learned, the official publication in the journal had been done today. So now it's official that also the latest amendments had been accepted and also been published. So they can be applied fully officially. Regarding the timelines and the requirements from the Article 5.5, again, to sum it up very briefly, on the one hand, we know that there are requirements which are already in place since 2022, so since the beginning or the end of the transition period of the IVDR, which means taking care of general safety and performance requirements. We know already about that. The transfer to other entities is not allowed, and also, of course, do not produce or manufacture your in-house devices on an industrial scale. What we also already know, and if we have a look onto the date one more time, 26th of May 2024 was the deadline for the major part of the requirements, and we see that is already in the past. So we are now already in a situation where all of these requirements, of course, could be checked by the competent authorities. Some of them are focusing on quality management aspects, and other ones are focusing on activities around these things and also creating a certain documentation. With today, I would like to focus mainly on the documentation aspects, so the creation of the in-house file that has been nicely presented also already by Professor Simon. So one more time, the Annex 1, this is a repetition, of course. We know that health institutions must ensure that the in-house IVDs are fully compliant with the GSPRs if they are applicable. We have the 3 chapters which can be found in the Annex 1 of the IVDR. The first chapter is dealing with the risk management topic. Second chapter with product-related features or characteristics like the performance and also aspects like electric current and so on. They are definitely relevant for in-house devices whenever there is such a characteristic available. And last but not least, the third chapter on the provided information which is supplied with the in-house device. Many of these might not be applicable, but still we need to have a look on them because they might be relevant whenever it comes to the safe and use application of in-house devices, and could be, for example, written down in operating instructions, SOPs, etc. Yeah, regarding the technical documentation itself, the in-house IVD file, we know that there is not an explicit definition what should go in there. We know that there are some certain requirements, for example, the conformity, the declaration of conformity, or the declaration, which is also published by the health institution, which describes that all of the GSPRs are fulfilled. This is an explicit requirement. However, many of these are not explicitly defined. But what we know is that the technical documentation must be drawn up in a shape or in a form that it can be suitable to enable the competent authorities, which needs to oversee the health institutions, to understand, identify, and also gain a nice overview of the in-house IVD in terms of especially safety and performance. That means that we need to have something in place in a written form to be able to demonstrate it. What is also important to mention here is that user laboratories should always check for national provisions, because there might be some national peculiarities, let's say, when it comes to registration. We have already seen that some countries are even asking for a pre-registration of in-house devices before the first usage. regarding notifications when it comes to problems with the in-house device. And last but not least, the language requirements, which also have been already mentioned, sometimes could be a national issue. So where to start from? I mean, we have the MDCG guidance document from 2023, which gives further hints on what the competent authorities might ask you for. We also have the Annex 2 of the IVDR. We know it's not mandatory, but we know it can be considered. At least it gives some insight in what a typical state-of-the-art manufacturer documentation would entail. But what we definitely see is what the typical sections within such an in-house IVD technical documentation should cover is, first of all, the identification of the device itself. Second, the intended purpose, which is really the core of the whole application, because it more or less determines what you have to prove and what can be cut out. This, of course, also concludes or entails the target patient group. We have seen that we also need to explain the classification, the risk classification that might influence parts of the documentation as well. We need to have a certain description of the in-house device. We need to describe the procedures and also aspects of the combinations. As we learned, there are different types of in-house devices. For example, repurposing RUO devices or setting up new combinations of not originally intended combinations of products by the manufacturers, etc. Of course, the general safety and performance requirements, we need to set up a checklist, a linked list. Risk management plays a major role in the GSPR section as well, and performance evaluation. So to validate and demonstrate that the product is fit for purpose at the end. For all of these aspects, you can watch our previous webinars. We had a lot of webinars so far on all of these different topics. And if you're interested in more details, I just want to encourage you to also watch our other sections. Well, the justification for the lack of equivalent CE-marked alternatives, we have seen that this is now again postponed for a couple of years. So the hard requirement comes up with end of December 2030, a little bit in the future. However, as we have seen, laboratories are anyways looking for potential equivalent market CE-marked devices on the market. Last but not least, the public declaration. Legally speaking, this is a very important document because it declares that the laboratory is really taking over the responsibility and also declares that they have done everything which is needed to make the in-house device or the LDT compliant. So where do we stand? Very briefly, all of these aspects now have been covered within the tool, as we have nicely seen in the presentation of Professor Simon. I can also just encourage you to have a look onto our website to see our wizard on strategy aspects. If you want to start with a huge amount of different devices, how to deal with that. And last but not least, also maybe good news for Professor Simon and other users. Very soon we will also have, of course, the declaration. So the only document so far which you created manually will, of course, also be covered in the application in the future. So to sum it up, how does our system work? I think the users already know. We have a certain walkthrough through all of the individual requirements, steps, and things that you need to manage and you need to do. The digital platform accompanies that on the one hand by providing information like our regulatory knowledge base, which is built into the point of care situation, let's say. We provide the templates in the product. We have the product database, which connects components which you might even request from manufacturers into your workflows. And last but not least, also come up with a certain standards database where you can easily identify potentially applicable standards for the individual challenges you have. This all is combined in the so-called Workflow Studio, so in the individual tools which build up the parts of the whole platform. And everything here helps you in saving a lot of time and guiding you through the process to set up finally your in-house IVD technical documentation. So with that, I would quickly jump into the application one more time to give you more insight and a certain feeling on how it looks like. So as you can see from top to bottom, this is the guide through the whole process. You already know that we start with asking a very general information on the in-house device itself. What you see here is a demo workflow which we build up for this presentation here in this webinar. Then it guides you through the whole intended purpose by asking you several questions and finally setting up the whole documentation, automatically pre-populates the stuff already so you can easily export it and handle it within your quality management system. The risk classification has been done already. Again, the same system. We have a classification wizard and a document which can be spit out of the system automatically after you have entered all of the relevant information. Within the Workflow Studio, you can build up a whole workflow if you want to define the workflow as being your in-house device, which is a very typical approach, I would say, in many applications, which makes this on the one hand complex, but on the other hand also very easy to be defined when it comes to the intended purpose for the device. The component utilization is a very important step where you can analyze the components used within the workflow and determine which type of in-house device you actually have created. For example, if you repurpose and research use only component to a diagnostic purpose, you exactly do that within the component utilization tool. Yeah, that leads to one of our largest modules, the General Safety and Performance Requirements, where you can enter the information, manage the information, and provide relevant evidence documents and determine if an application or if a General Safety and Performance Requirement is applicable or not. So the tool also helps you in determining this status. The user environment helps you in describing the environmental conditions which sometimes are needed, for example, when it comes to storage of some devices. So you can draw up even this part of the documentation in a very streamlined and easy way. Performance evaluation, here we provide some templates to set up a plan. Typically, you need to determine what or which parameters you want to validate and how to set it up, sample size, etc. And of course, to document your reports, to document your results in the evaluation report. Then, as we have seen, we have a large module on risk management, setting up a plan, setting up the records, and setting up the report. All can be done in the form of wizards, but also in a studio which even allows you to manage your individual risks very nicely via a Kanban process. Yeah, last but not least, another module which might be released very soon is the user information, again with a wizard, which even helps you in determining the accuracy of certain labels, instructions for your medical device, and even helps you especially with the chapter 3 of the GSPR topic. Yeah, and when you're done with the whole process, then we have another document, as I already indicated, which is the declaration, which is a new part here. And typically the document template looks like that. And again, as you can see, many of these things have been already pre-filled by the tool. So first of all, the identification of the health institution as required, the identification of the product itself as requirement, the classification. And here you can also discuss within the document the needed information about the fulfillment of applicable GSPRs. Normally or typically you fulfill all of the applicable GSPRs. However, the MDCG document or guidance document also allows you to discuss some of the GSPRs, and also if that is the case, then you need to justify it and also declare it publicly. However, this document is also automatically set up very easily, and if you then complete the document, you receive the final document. You also tick it off with that. That means document is done, and you can export it and again put it into your document shelf or publish it on your website or whatever your preferred way to present this to the public would be. Yes, and with that, I think we have now demonstrated that the whole process of the setting up and creation of the documentation according to the requirements from the IVDR is possible now in the tool and also be used already a lot by our customers. And with that, I would like to hand over back to Simone. Thank you very much for the attention and open to any questions.
Thank you again, Andreas, and the guest speakers for this interesting contribution to our IVD Ready to Go webinar. With this, I would like to briefly summarize what happened during the last year. The journey started over a year ago, where we kicked off the webinar series. It was a German kickoff webinar featured by IVDR expert Dr. Sonja Neuhofen, who gave an excellent first overview. Our second webinar featured expert Dr. Christiane Gebhardt, who gave an excellent view on making choices in relation to the Article 5.5. Here we described how, using a risk-based assessment, the amount of documentation can be determined. In webinar 2, it was about the intent and purpose for your in-house tests, followed by webinar 3 on the knowledge based on our platform and ISO standards. Finally, webinar 4 focused on the documentation strategy in November. The topic of setting up a correct intended use seems trivial, but it's all but easy upon closer inspection. The opening of the Gates event in November was a milestone for us at Platomics, bringing much of the work that we did in the background out to the laboratory users. These webinars featured a number of experts from the field, including Daniel Hübschmann and Peter Horak from the DKFZ and NCT in Heidelberg. From the regulatory field, besides Christiane Gebhardt, Erich Zeisel and Marisa Lien shared their professional insights. Webinar 5 was the webinar with the most commented title, Standards Are Beautiful. Here, Professor Satlokal talked about standardization and sample quality, showing how applying standards leads to better results using pre-analytics actually as an example. Professor Satlokal also touched upon the importance of ISO 15189 for a lab. Here, Platomics released the GSBR Wizard and the GSBR Studio that was presented by Professor Ronald Simon and Andreas, of course, today. Webinar 6, entitled An Ounce of Validation, featured by our own Andreas, was joined by 2 more colleagues from Platomics, Clara and Karin, talking about performance evaluation. Here we presented our NGS validation service for genetic laboratories. The most recent webinar on risk management showed us joined by Professor van Stapel, who discussed risk management. Professor van Stapel was very clear that the IVDR is here to stay, but also that it will rid the market of many substandard devices, thereby benefiting patient health in general. It also offered the audience a view into our risk management studio that we developed, which is one of the last major modules in our program. Which brings me to today. May 26th has come and gone, and the next phase of the progressive rollout is live. We are IVD Ready, and we welcome you to join us. Our TechDoc and GSBRs are IVing documents which will require a constant degree of updating and maintenance, not because of the progressive rollout of the IVDR, but because behind every sample is a patient. New technologies will be introduced and new treatments are heading our way. So let's see what else is coming our way. All webinars and the recordings are available free of charge on our homepage, and you can find them also on our YouTube channel. The recording of today's webinar will be available on our website by next week. When we look at our Plato X assistant, we will keep making improvements. It will become easier to put everything together and work towards your declaration of use. Furthermore, we develop new features. For example, for the review, experience gained from clinical use of your in-house tests are planned. Of course, we also need to expand the languages in the software, as we fully intend to have everyone in Europe using Plato X IVD Assistant. So besides English and German, the assistant will be available in other languages too soon. With this, I want to express my sincere thanks to all the participants who have joined us in our program IVD Ready by May 2024 from around Europe. I would like to thank the guest speakers for their highly valued contribution to the IVD Ready webinar series, and Andreas Oberleitner for his contribution as well, and for demonstrating the Plato X features. Of course, I thank the whole Platomics team. It was an amazing and busy year. Thank you for your attendance. Stay tuned, and I wish you a pleasant afternoon.